Arizona State University and Science are recognizing researchers who address pressing problems. The ASU Science Prize for Transformational Impact honors early career scientists developing innovative solutions to today's biggest challenges. Hear from the inaugural winners on ASU's Lab Coat Optional podcast and visit science.org/ASU to apply. This podcast is supported by the Icahn School of Medicine at Mount Sinai, an international leader in research, education, and patient care. The school is the academic arm of the 7-hospital Mount Sinai Health System in New York City. It is consistently ranked among the top recipients of NIH funding. What are researchers in genomic medicine working on to transform patient care and improve patient health? Find out in a special supplement to Science Magazine prepared by the Icahn School of Medicine at Mount Sinai in partnership with Science. Visit our website at www.science.org and search for Frontiers of Medical Research-Genomic Medicine. The Icahn School of Medicine at Mount Sinai. We find a way. This is a Science Podcast for July 23rd, 2026. I'm Sarah Crespi. First this week, freelance science writer Brendan Burrell talks about a previously unreported death in a gene editing trial in China. After that, producer Megan Cantwell talks with researcher Evan McKenzie about the secrets of an insect that migrates hundreds of meters underwater. Now we have freelance writer Brendan Burrell. He wrote this week for Science and Retraction Watch a story on a death in a gene editing trial of one. This has not been publicized up until this point, but in spring 2025, a 6-year-old girl in China died after receiving gene editing treatment with a base editor. We'll get into what base editors are in a little bit. Her parents came forward and have asked to remain anonymous. So we won't be using names for the parents or the child in this segment. Hi, Brendan. Welcome to the podcast.
Hey, thanks for having me, Sarah.
Can you talk a little bit about how the parents entered this situation? So it's parents looking to get treatment for their child. They find a researcher. How did that happen?
These parents, they had this young girl who was starting to fall behind her peers in kindergarten. One teacher suggested that she get an evaluation from some experts. They learned that she had global developmental delay, which is kind of a general term, but they wanted to find out kind of what the genetic basis of that is. And eventually they learned that it was a mutation in a single base in a single gene that affected how her brain developed. And they by then had joined sort of a online group where parents traded advice about their children who had various neurodevelopmental disorders. And someone shared a talk by a researcher named Zulong Cho. And he was kind of a rising star. He was a geneticist who'd done work in a lab in the United States. He'd gone back and he was developing some really cutting edge gene therapies.
And they reached out to him directly and said, what about this gene? It's only one base, right? We know what our daughter's genetic condition is. Is there anything you can do about it?
Yeah, I mean, they sent him their report of their daughter's genome. And he looked at it and he saw that this was a really promising target. This form of editing, base editing, had started to be used, but nobody had targeted the brain in a human yet. So that was kind of the next frontier. And so he invited the family in, they sat down, they talked. And I think the key thing here is the family said, we're willing to fund this. If you're a researcher, that's obviously really helpful. I mean, no matter where you live, whether you're in the US or in China, having a source of funding is always a challenge.
How much money would something like that cost?
Depends on how things progress. Over there, doing the preclinical work and starting the clinical trial is around $1 million. I don't think the family initially knew how big of an investment was going to be. They wanted to just start going in that direction so that their daughter had a chance. They were willing to do anything for her.
So how did you first come across this story?
Retraction Watch received an email from the family at the center of the story. They were really upset by how everything played out. They had lost their daughter in this clinical trial. They didn't feel like there had been accountability. So they reached out to the journal where the study was published, Nature, and they sent an email to us in the end.
So Retraction Watch is kind of famous for examining this intersection between what goes on in the research world, what goes on in the publishing world, and how when mistakes are made, how it's handled in those two different places. When the parents reached out to Retraction Watch, what do they want? What were they looking for from this?
It was kind of surprising that they were reaching out to Retraction Watch, but initially, like the issue that they were focused on was they felt like the study itself should be retracted because they didn't feel like it fully disclosed what had happened. The study, just to be clear, was of the animal, the preclinical results. There was nothing about the clinical trial. There was nothing public about it. And they felt like that needed to be corrected somehow. And as I started talking to them, I started to learn more about the story and talking to other experts, realized there are actually some fundamental concerns with how the clinical trial had been conducted that I started digging into.
This is a, I guess you'd say, personalized gene editing experiment where it's one person, one specific mutation that the researcher is trying to address, and it's all bespoke, but also somehow a research effort as well as basically an attempt at treatment, right?
That's right. These kinds of projects are kind of a sprint, and especially in this case, the young girl had a neurodevelopmental disorder. She was 4 years old when the family first approached the researcher, and they're kind of running against the clock because you want to fix something like that while the brain is still developing. Of course, before you put something into a person, you've got to test it in mice and monkeys. So they were moving fast.
How different is approval of these kinds of trials, clinical work in China compared with the US? I'm pretty familiar with the different kinds of stages of trials here.
Well, I think to some degree, China has tried to model their system after the system we use in the West, but they've had a few stumbles along the way. There was a big controversy around gene edited babies a few years ago because that was considered an ethical no no.
There was a lot of scandal here abroad, and basically, didn't he end up getting punished in some way, the researcher?
He did go to jail. I can say that for sure. Basically, I think the Chinese learned that there were also their system had some gaps and reforms were needed. And so they started overhauling how research and clinical trials were conducted. And generally, especially with like a new therapy, a gene therapy, you need to get approval from basically China's equivalent of the FDA, and then also register with a health body that oversees clinical trials and get ethical board approval from your local institution.
So that sounds pretty similar.
It's pretty similar, but as we learn in the story, what that means exactly in practice is a little different.
Between the preclinical and the clinical trial, we're talking about almost $1 million. My understanding is that the preclinical steps are figure out what the change in the gene does to the protein, figure out what that looks like in a mouse. So you put the change in the mouse gene and express it in the mouse and see what happens. And then how does the mouse respond to a base editor that has to be designed specifically to correct this mutation? And then can you get that base editor into monkeys to see if it can edit the genes inside of neurons inside of monkey brains? That's kind of the preclinical steps that they need to go through.
There's a final step there, which is you also want to test for toxicology. So that's a separate set of experiments.
That does sound expensive.
It is.
There's a couple other proof of concept things going on in here. For example, like gene editing neurons in the brain, getting the vector into the brain. Also, this is a base editor that's slightly different than CRISPR, which I think a lot of people are familiar with. Can you kind of talk about how it's different?
Yeah, I mean, CRISPR is kind of the original, and it involves basically cutting the strand of DNA in half and then repairing it that way, which can kind of lead to some weird surprises. The base editor only cuts a single strand at a single base, and then it replaces or it chemically changes that base. And then the cell's natural repair mechanisms will repair that. It's a very precise way to fix just one base if that's what the problem is. I mean, it has its own challenges. It's not 100% perfect, but it is actually easier to control, which makes it really promising as a therapy.
Has that been used in people before?
Well, yeah. I mean, in fact, while this Chinese trial was just getting underway, there was quietly another clinical trial taking place here in the US. It was the case of baby KJ at the Children's Hospital in Philadelphia. KJ had a metabolic disorder at infancy that was potentially fatal. And there was a sprint to repair that base. That study was published. It reported a remarkable success. But at this time, that still wasn't known. This was still just sort of a, uncharted frontier, and they were going for the brain.
So they were using a base editor. This Chinese trial was using a base editor, but the targets were different. That was liver, so it's a little bit less difficult to access. And then also, there wasn't a virus vector. That was another one of the differences. Can you talk about how they delivered this base editor to the brain?
The delivery has always been a challenge for gene therapy. I mean, the earliest gene therapy studies did use these viruses known as adeno-associated viruses. You inject it and it gets into the cells and then starts producing a response.
Yeah, so in this case, the base editor was too large to fit into a single viral vector.
Yeah, I mean, you've got to have the base editor and then you've got to have the actual instructions. And these adeno-associated viruses don't have very big.
They're tiny.
Yeah, they're tiny. So they had to split it into two, which was potentially a viable strategy. I think when I talked to experts, they were somewhat divided on whether that was a good idea or not.
You're trying that, plus you're also trying to get it in the brain. And we talk about the brain as kind of difficult to target because it has extra barriers. And also, it's very difficult to see what's happening in real time with cells in the brain. What were the expectations of these parents when they decided to go ahead with this treatment? What did they think was going to happen?
They were not too worried. In fact, the biggest issue was the therapy may or may not work. That's what they were told. They were told again and again, yeah, there can be some adverse reactions, but everything was looking good.
You have from the parents here recordings of their interactions with the research team describing the treatment and the risks. And how did that help you in your reporting?
One of the things that was really remarkable about this and helpful about this story is this family, they weren't scientists themselves, and they started recording their conversations with the researchers and doctors so they could review them later. They ended up sharing that with me, and it helped me understand what the researchers were telling the family, what the family expected, and allowed me to really reconstruct things beyond what I saw in the documents that the family had also shared.
So, like informed consent documents that can read kind of long and technical or like spread out over months.
Yes, the informed consent document can be just a full inventory of everything that's possibly happened. But then you sit down with the doctor and they say, don't worry about a thing. And that's kind of what happened in this case. Explicitly, one of the concerns that people here raised was the document did not mention anything about possible death, or it said that these are some serious consequences that must be treated quickly. And then in person during the conversation with the lead medical doctor, he basically said he wasn't concerned about anything.
So they didn't mention death in the informed consent as a possibility.
No, it was not in there.
This is not a fatal disease that they're trying to treat here. Like with KJ, like it was a race against the clock because this is something that could possibly kill him. In this trial, this little girl had a developmental delay that wasn't predicted to be degenerative, right?
This condition that she had a pretty wide spectrum from mild to pretty severe. And she was on the more mild end of it. And she probably could have lived a decent life. That's something that the family has struggled with in terms of this decision they made. I mean, they loved her so much and they wanted her to live a normal life. And they took a risk that they didn't realize they were taking.
Can you talk about what happened during the infusion and, how she died?
So they go in and she receives the infusion. Basically, the infusion goes through into the spinal canal at the base of the back, and then the idea is that it will disperse into the brain. After a couple of days, she starts to get a fever. This is a normal reaction to a viral gene therapy. It's just the immune reaction, essentially. And then that starts to go away. But actually, something else is happening, something more troubling. To the girl, she hasn't urinated since she's received the injection. The doctors have to stop giving her any fluid. Her kidneys are shutting down, and they transfer her to the intensive care unit. She ends up dying 7 days after receiving the infusion. The final autopsy report indicated that she had a condition called thrombotic microangiopathy. It's like this pattern of clots in the kidneys, and that is a known reaction to high doses of these viral gene therapies.
Do we know what might have gone wrong here? Like, did she get a lot of virus more than maybe in past gene editing experiments? Or, is this just so different because it's in the spinal column aiming for the brain? Can we know, what happened there?
We don't know the exact dose she received. The family does not have a record of that. What we can say is that when this drug was given to monkeys, they did a high dose and a low dose. And in both cases, there was some pretty severe liver damage. And there was some indication that there could have been the same problem in the monkeys. But, an expert I spoke to, James Wilson, a famous gene therapy guy, he said there should have been more primate study. They should have been looking at the acute response in the month after the infusion and so on and so forth to actually figure out what is a safe dose. And it does not appear like they did that.
What kind of triggered the parents coming to retraction watch was that they didn't see consequences to this. What did happen after her death?
What happened was the hospital received a small fine from like the local health authority, and that was about it. The family asked the principal investigator, they actually didn't want their information about their daughter to be published. That's sort of the way it seemed like things were going. But then about a year later, this paper appears in Nature. It basically has all of the animal data and it suggests that, oh, this is a promising strategy for a potential therapy for people. No mention that actually we tried this and it didn't work. So that was very upsetting to them. Number one, they didn't feel like the lead researcher should get kind of the plaudits that come with a Nature paper. And number two, they felt like people who read that might be misled. And in fact, within their parenting group, some parents were very enthusiastic when they saw the paper as, hey, this is promising.
When you talk to experts about what showed up in the paper, as opposed to like what the parents heard from the researchers in the moment, or what do people looking at this paper say about it who are experts in the area?
They raised some concerns about some of the images and some of the data in the paper. They felt like, the effectiveness of these adeno associated viruses is typically much lower than the authors had claimed. They had some concerns about whether it was really as effective as the paper made it appear.
And there's also a question of the funding because the parents paid a lot of money towards all of this to happen for their daughter. And then they're not mentioned in the paper as funders.
I have to say, I mean, the lead investigator was kind of in a challenging spot, right? He has this family that says, we don't want you to publish information about our daughter. And in fact, this is something I discovered between the first submission of the paper and the publication of the paper. Actually, the monkey and mouse data was taken out, and they appear to have redone all those experiments. They still use the same mutation that the family's daughter has. This is a therapy targeted towards that mutation. I mean, you should probably still thank a family for their assistance because they were the catalyst for this whole thing. And certainly there should have probably been some revisions to warn about in some general way.
Yeah, the safety data, particularly, right?
Yeah, yeah.
When you were working on this story, you must have reached out to the lead researcher, Cho. What was his response?
I didn't get a response from him. I reached out to him several ways. I reached out to him on WeChat, which is a Chinese social network, and to his personal email, and I got nothing.
And I know you haven't heard from Nature yet, but they did respond to something the parents wrote.
That's right. The parents sent a complaint to Nature's ethics team. Nature said that the issues they raised fall outside their purview in terms of data integrity and directed the family to contact the university. I mean, at that point, the family had already reached out to the university and was basically told that the university did not believe these were ethical issues.
After this interview, actually, Brendan did get a reply from Nature stating that the journal was not aware of the issues surrounding the clinical trial, but that when concerns are raised about its research, it will always look into them carefully. And then what about the Chinese version of the FDA, talking to regulators or talking to lawmakers? What happened here? Is there any response from them?
I don't know if the family reached out to the regulators in any way. And the death of this girl has never been made public before.
Wow. You consulted ethics experts for this story. Did they see problems with either the way this was conducted, the way that it got published? What are their primary concerns?
One of the biggest ethical concerns with the trial was the researchers did have some warning that there were some toxic effects in monkeys. Turns out that study was never shared with the ethics board of the hospital. And the principal investigator had told the family that it was nothing to be concerned about. So that's like probably one of the biggest red flags. Some of the experts I talked to said that when a family funds a work like that, that should certainly be disclosed. There was a sense that the family was pressured to contribute more and more funding. There were a lot of informal negotiations that took place. There were gifts that they felt encouraged to give to the principal investigator. And the family, I mean, they'd started the ball rolling with this research and they didn't feel like they could stop it. They had to keep paying because there was nowhere else to turn. That was a major concern as well for this type of research in general. In many cases, an N of one clinical trial might be funded by foundation or a research grant. But I think increasingly we're going to see studies like this funded by the parents themselves. And it's critical that there's some kind of firewall there to protect the families.
You talked about like how the parents, felt that the publication was problematic, that there weren't enough consequences for what happened. But, how are they actually handling this? Like, you talk to them, what's, yeah, I just can't imagine what it's been like.
They are absolutely devastated. I mean, when this happened, they left their apartment where their daughter's things were, and they are living in a new place. They barely talk to each other about the death. The mother has not gone back to work. They've cut off contact with many of their friends, other parents, because they just don't want to face it. I had a lot of conversations with them that were pretty difficult.
In 1999, there was a death in a gene editing trial. This was Jesse Gelsinger. This was the first death in these types of trial that was reported. It put a freeze on genetic manipulation of patients for a while, and the consequences for the researcher and the institution were pretty dire.
The university received a major fine. Jim Wilson was barred from human research for 5 years. There was a lot of media coverage of it. So far, that hasn't happened with this case. Like I said, the death has been kept quiet. My hope is that when this story comes out, there might be some reflection on that.
Brendan Burrell is a freelance science journalist based in Los Angeles. This story was produced in collaboration with Retraction Watch and supported by the Science Fund for Investigative Reporting. You can read the entire story at science.org/podcast. Also out on the site this week, we have a story by news editor Tim Appenzeller on the first contagious fish cancer. Transmissible cancers have been seen before in Tasmanian devils, where they show up as a facial cancer that goes from devil to devil through bites. And now genetic studies have shown that these catfish have melanomas that are a single clone. Really interesting discovery there. In the book section, we have a review of the book Snake Men, Rebels, Reptiles, and the Race to Name the Creatures of Earth by Zach St. George. In his review, Christopher Kemp writes, this book is about the tension between the slow, scholarly pursuit of taxonomy performed in institutions and museum archives and the democratic, less policed version of species description being conducted in basements by upstarts and unaffiliated amateur scientists. From the journals, we have a package of papers in Science and Science Advances coming out this week about the 4D genome. As the intro says, the human genome is not a static list of instructions, but a dynamic, three-dimensional structure that folds, loops, and reorganizes within the nucleus to control when and where genes are expressed. This spatial and temporal organization, the 4D nucleome, has emerged as a critical layer of gene regulation, one whose disruption is increasingly linked to disease. So lots to look at there. Okay, back to the main show. Next up, producer Megan Cantwell talks with researcher Evan McKenzie about an insect that has a respiratory system able to withstand high pressure at depth.
Insects occupy almost every ecosystem on Earth, but not the open ocean. Tiny animals like zooplankton survive in the ocean by feeding at the surface and then descending deeper to avoid predators. And some researchers think that a major reason that insects haven't been able to do this is because if they underwent a similar migration, their respiratory system would not be able to handle those high pressures. This week in science, Evan McKenzie and colleagues show how an insect in Malawi has adapted to dive deep into a lake. Thank you so much for joining me, Evan.
Thank you for having me on.
So I wanted to start with just talking about this insect. Can you talk about what does its larvae look like? What does it look like in adult form?
The Latin name is Chaeoborus edulis. So Chaeoborus is the genus, and there are lots of species of that all over the world. And edulis, species name, it means edible. It's the edible Chaeoborus.
Oh, right, because I looked it up and they're like in mud cakes or something.
Yeah, so when they emerge as adults from the surface of the lake, it often happens in these really dense clouds, which can blow ashore. And then people with sort of a greased pan inside of a basket swing it around in the air to collect them. And then they can compress them into a cake called kungu cake. Kungu is the local Malawi word for the lake fly. As the adult form, they kind of look like mosquitoes. In the case of this specific species from Malawi, they're a little bit smaller, hairier antennae, and luckily for us, they don't have biting mouth parts. However, they spend most of their lives as the larva. They're basically a transparent tube almost, where the only non-transparent parts are their eyes, as well as the two sets of buoyancy control organs or air sacs, which is what I was specifically studying. They progress through 4 larval stages, which takes the majority of that 3-month period. Then they become a pupa, still in the water, before gradually coming up to the surface to metamorphose into their adult form, fly around, mate, hopefully, and then die. Its pressure tolerance is a little bit less at the early instar stages and gradually increases as they develop.
So tiny and transparent helps if you're looking at it under a microscope, seeing its developmental process, but not as helpful if you're trying to actually observe it in water. They've previously been observed at different depths, right? But in your study, how were you able to actually see this migration?
So to look at the details of their vertical migration, we used a sonar system that we were able to deploy to the bottom of the lake such that the transducers were looking upwards in water about 300 meters deep. The air sacs or buoyancy organs contained within their body are also very good for scattering sound waves, which is what the sonar relied on to show us where they were. We got a much better idea of the timing of their movement pattern for sure. So they begin their dive from the surface in the very early morning hours because they need to go quite deep. This takes a while. They're very tiny larvae. It confirmed for us that they reach a level between 250 or 230 meters below the surface, approximately, which is the same depth at which the chemistry of the lake water suddenly changes to be almost totally lacking in oxygen, which prevents their fish predators from following them into that zone. They stay down there all of the daylight hours, as well as the time required to traverse the distance between the surface and that depth.
So your study is focusing exactly on how their organs are responding to these pressure changes. What were you observing at these different pressures?
We wanted to have a look at how their air sacs respond to hydrostatic pressure. So a lot of people are aware of how it feels when you dive very deeply in a pool and can feel that pressure on your ears, even after only a few meters. But with these larvae diving 250 meters, that actually adds the equivalent of 25 additional atmospheres worth of pressure onto these very small air-filled containers. So the material that the wall is made of has to be able to withstand all of that pressure that's not inflated from within like a balloon or anything like that. So we wanted to understand the material properties that allow them not only to resist collapsing under those pressures, which would be the end of a larva if that happened, but also push back sometimes against that pressure.
So the air sacs are the part of the organ that's controlling how it's kind of going up and down. So how does that relate to like their respiratory system, I guess? What is sort of the internal structure of these larvae?
Air sacs are derived from the respiratory system. However, they no longer function to exchange gases with the environment at all. So they're entirely sealed within the body, and they're modified to contain 2 different types of material. One is the sort of stiff, inert, unresponsive cuticle that is the same material that tracheal systems more generally are made of. And the other material is sort of sandwiched in layers between layers of cuticle. This is called resolin. It's a very interesting insect protein found in soft and flexible regions of exoskeleton. And it's what's responsible for allowing the air sacs to change their volume and have a little bit of elasticity.
What was your experimental setup here to actually see how these air sacs are responding to different pressures under a microscope?
So it's really quite a simple setup. We just had a hand crank and a digital pressure meter attached to the system. So we would by hand go up, increase the pressure by increments, and take images just using a USB camera attached to my laptop in order to collect the data.
The supplemental videos show the air sacs actually imploding. And it's just crazy because it's like, one frame there, and then it's just like completely burst the next, which I think is pretty cool to see.
The pressures are so great that, yeah, it does sort of result in a very dramatic shattering of the air sacs. And I don't know if you saw both videos. There's the one that shatters, but it also kind of depends on the pH of the solution that the air sacs are in. There's one that's in a more alkaline pH, which instead of shattering, you can see the air pocket inside the air sac kind of just suddenly disappear, and then the air sac expands to the size that it would have been without pressure.
So it's not just pressure change that they're responding to. Internally, they're also able to control their buoyancy by actually changing the pH in their body. How exactly is this working?
So the pH of the external environment is fairly stable, but what the larva does is regulate the pH of the air sac itself, and that is what regulates the size of the air-filled organ. When the larva engages processes which increase the pH of the air sac, that causes the flexible protein component of the air sacs to begin swelling and taking on water. And this is a totally reversible process, so they can then choose to decrease the pH again if they want the air sacs to shrink, causing the larva to sink again.
And so this pH control that they're doing, are they constantly doing this at greater depths too?
We haven't really been able to observe what they're doing in the natural environment with like a pH meter in their body cavity or anything like that. Based on experiments that we did, where we specifically applied different pHs to the air sacs and also hydrostatic pressure, increasing the pH of the air sac is really only useful to the larva within the top 50 meters or so of depth. And beyond that, the pH-based buoyancy regulation mechanism has sort of reached its limit. And instead of relying on that mechanochemical means of regulation, it seems that the layers have all kind of cinched up and are now meeting each other, sort of enforcing a minimum volume so that they still have this, like a water wing, basically, allowing them to not sink, which is an advantage over other things like copepods and Daphnia, etc.
That's so cool that they're able to tune that. You didn't just look at this one species, you looked at other species within the same genus that are found in different lakes that aren't as deep. Are their air sacs different than the ones you see in this lake in Malawi?
We very much did see differences. In Lake Malawi, the lake is actually much deeper than 250 meters, but they go down to that specific depth because that's how they escape from fish. So in Lake Victoria, where two other species actually coexist, they go all the way down into the sediment of the lake, which is approximately 80 meters. And the maximum pressure withstood by their air sacs is only a little bit more than the pressure resulting from 80 meters. Even lesser still is the pressure resistance of larvae that for a longer time I've been working with, which we can find in lakes near University of British Columbia here in approximately 20 meters deep. It does seem that shallower dwelling species have a combination of thinner walls, but also larger radii or widths to the air sacs. So an important principle that many engineering folks probably know is that a pipe under pressure, be it pressure from within or without, all else held equal, is going to be better at resisting that pressure if it is narrower. And so it seems like the larvae have taken advantage of that principle.
What do you think the findings for your research mean for why we don't see insects in the open ocean yet if this tracheal system crushing and imploding at deeper depths might not quite be the reason since we see it in this species?
Even though this is just one species and we're not seeing it in other groups of insects, insects are famous for having all sorts of different weird and interesting and very neat adaptations. Evolution can sometimes act very rapidly upon them. And in fact, Lake Malawi hasn't even been as deep as it is for all that long, at least in terms of evolutionary or geological time. If the reason that there are no insects in the ocean truly was this dive limit based on their tracheal system, then surely something like Chaeoborus would have invaded something like the Black Sea, which has actually a very similar zone of anoxia to Lake Malawi.
What else could maybe be underlying why they haven't colonized?
I should say this is pure speculation, and someone smarter than me in this area may disagree, but I would be tempted to lean towards an explanation regarding the osmotic challenges of seawater and other sort of chemical environmental factors which just don't seem to play nice with insect physiology more specifically. There is also the hypothesis that, the niches are filled with things that are very similar to insects, crustaceans already. However, insects invade new niches all the time, which aren't necessarily empty. So I don't know. That doesn't quite completely do it for me.
Well, thank you so much for taking the time to speak with me about this. I appreciate it.
Thank you very much.
Evan McKenzie just recently completed his PhD and is a researcher in the zoology department at the University of British Columbia. You can find a link to his paper at science.org/podcasts.
And that concludes this edition of the Science Podcast. If you have any comments or suggestions, write to us at sciencepodcast@aaas.org. To find us on podcast apps, search for Science Magazine, or you can listen on our website, science.org/podcast. This show was edited by me, Sarah Crespi, Megan Cantwell, and Kevin McLean. We had production help from Podigy. Our music is by Jeffrey Cook and Wen Koi Wen. On behalf of Science and its publisher, AAAS, thanks for joining us.
Best thing that's ever happened to you financially. Go.
Easy. Sold my car on Carvana. Amazing offer.
Really? I hit 200 on the scratcher.
Did the scratcher come to your house and hand you a check?
No.
How many scratchers did you hit to get that? I hit a button on Carvana.com once.
Okay, that's fair.
It's like the lottery, except you always win.
Not like the lottery at all, actually.
Exactly. Inexplicably good. Offers worth bragging about. Sell your car today on Carvana. Pickup fees may apply.